Owen. Welcome to the Seattle podcast.
Thank
Thank you
you.
so
So glad to be
much.
here.
Yes. Okay, so to everyone else, you're Dr. Owen Richfield.
hmm
You're Owen.
I am Owen. I am Owen. Yeah.
You
If you
are.
know me, my
Okay.
name is Owen. Yeah.
Right. Dude, you are amazing. You are a PhD biomedical engineer.
Yep.
Tell me
Yep.
tell me how you're going to change the world, Owen. What's your mission in one sentence?
Yeah. So my main mission is to eradicate kidney disease. That's my main mission, number one.
Yeah, not too big at all.
Yeah. Yeah. Not a big deal. No big deal.
Okay, you've done so many things that is leading up to eradicating this kidney disease, and it's huge. And we'll talk more about the tools in itself. But there are three things that we're going to talk about.
Yeah.
One is your publications on kidney and the drug delivery, which is pretty extensive. The second thing is you are at Yale and with the Saltzman lab, and then digital tools in biomedical industry. So let's start with how we got to the publications, because you have written like way too many papers.
Not enough, really.
Not enough. So first, tell me why you're so obsessed with kidneys, and then contextually.
Yeah, absolutely. So, firstly, my excitement with kidneys, I really got started in the kidney space when I met my PhD advisor. So, I did my PhD at Tulane University in New Orleans. It's also where I did my undergrad. So, at that point, I was an undergrad at Tulane. I was looking at a PhD program, and I met Gabby Navarre. And Gabby Navarre has been working in the kidney disease research space for 50 years.
That's
He's
forever.
seen everything,
Wow.
he's met everyone. Many of the people that he taught are now retired, and he's not. So he's really a special guy. And as we were talking about the kidney, and he was sort of introducing me, really, I knew so very little about the kidney at that point. What I realized is that there's a lot of math in the kidney,
Hmm.
and I'm a math guy. I was a math major. I've always been, interested in numbers, in ways that we can use numbers for important, real tasks that we need to perform in our lives. And there's a lot of math in the kidney. There's a lot of math to figure out how the kidney is doing. If the kidney is in good health or if it's in bad health. And the math just keeps going deeper and deeper. And so Gabby, by the way, his full name is Luis Gabriel Navarre. Everybody knows him as Gabby personally. So that's why I call him Gabby. But of course, when I
Got
was a
it.
student, I called him Dr. Navarre. But the point being that once I got introduced to it by him, it just kept going deeper and deeper. And just from an academic standpoint, I was very interested in the kidney. My grandfather passed after his kidneys failed after a surgery. My dad had kidney injury after a surgery. So there is a personal component, for sure. And it is, and I've met kidney patients. I've met people with varying degrees of kidney disease, and I am absolutely passionate about helping people with kidney disease. That said, that is one of the motivations. Another motivation is just honest, pure interest. I'm just interested in it, and I find it to be very interesting and exciting.
Okay, can I ask? This may feel like an outlier question. So there's
Sure.
like so many other organs,
Yeah,
and that I'm sure requires other math too, lots of
totally.
science, lots of integrity in those areas.
Yeah.
And there's like whether it's heart or other things. What
Yeah.
made you be like, kidneys, this is it. This is where I'm gonna go all in.
Totally. Yeah. So that was its own kind of decision. Because there were opportunities for me to look at the liver. There were opportunities for me to look at the lung. There were opportunities for me to look at the heart for sure. So there's a lot of math in heart. And so I know a lot of people that were saying, hey, if you want to get more grants, or if you want to if you want to, have more kind of excitement around your work, maybe try a sexier organ. And I know that sounds like a really funny thing, but actually biomedical people, they're very excited about the heart and the brain. Those are like the main organs, right? And then after COVID, the lung became more popular. But
Interesting.
yeah,
Yeah.
but the thing about the kidney is that one, kidney disease is on the rise in a way that's worse so than many other organs in the body. It's also less understood. It's also less thought about. So when we think about niches, like in the academic world, kidney is actually a better bet because there's fewer people looking at the kidney. And the people that are looking at the kidney are often very supportive of other people being involved, so long as everybody keeps their own space to themselves. But if in the end the kidney has, I think, a lot of opportunities, there's just so much we don't know about the kidney. And so that, for me, that was a big driver beyond my personal interest in it. It was actually, there was a strategic aspect to it, which is that, there's opportunities in the kidney space that there are not elsewhere.
So this is what makes you really a scientist, because you're pioneering through
Fine.
uncharted territory.
That's the goal. That's the goal. Yeah.
Got it. Okay, that makes total sense. But also, like, even if it's not the heart or the brain or lungs, they're also interrelated in some shape or form. So you still have to study all of that because
Yeah.
it's all affected, right?
Yeah, and I think actually for the kidney especially. So like the kidney connects the heart to the lungs in a lot of ways. And I'm not just talking anatomy wise, I'm talking physiology wise. So really when you look at the kidney, you have to look at everything. It's just that my focus has been specifically on what's going on inside the kidney. But in reality, whatever happens inside the kidney has effects on the rest of the body. And that's what we see. I mean, these days, so many people are dying of heart disease, but actually, a lot of the time, they also have kidney disease. And that's why their heart disease got so
At
bad
the same time.
at the same time. Yeah.
Oh,
Yeah.
dang.
Yeah. And vice versa. So if you have heart disease, you're more likely to have kidney disease. If you have kidney disease, you're more likely to have heart disease.
Oh, can't escape from either then. Okay,
No.
if you're in it.
They're
Got
yeah,
it.
they're very interrelated. So, and I mean the same thing is that if you have a lung condition, you should be checking your kidneys also. Because there can be if the lung is damaged or if there's a problem with the lung, the kidney may have to work harder and then may get injured in that process.
That sounds extremely complicated, and
Yeah.
it sounds complicated because it's like, oh, okay, if you find out that you have kidney disease, it's like, okay, you still have to think about heart disease, which is like extremely complicated. And you're like, okay, what kind of surgery do I have to do?
Right.
And who do I have to talk to? Who are all these specialists? And then not even talking about like the drugs in itself and what's in clinical trial or not, and what are
Yeah.
your chances, right, of being
Yeah.
able to extend and prolong your life. Is that right?
Yep. Yeah, absolutely. So I think a really good example. So a lot of the time, so a lot of people, especially in America, but across the world, have high blood pressure. This is very common, right? High blood pressure, the way that, and the concern about high blood pressure is that it will hurt your heart. So pretty much like, it'll cause atherosclerosis and then you could have a stroke. So it would get your brain. Or you have high blood pressure and that hits your heart and eventually over time that can damage the heart to then the point where vessels get clogged and you have a cardiac arrest. So in both cases, though, when you have high blood pressure, the drug that you take, those drugs act in the kidney. So it's actually the kidney
Oh,
that is
really?
being changed, and then that's what protects your heart and your brain.
You
So,
know, that
yeah.
is, I did not know
Yeah.
that, Owen.
Oh yeah, big time. I mean, I'm talking like
Oh,
if your dad has high blood pressure,
yeah,
he probably
yeah.
takes a drug that acts in his kidney because that's where those drugs act.
That's wild.
Yeah,
I learned
totally.
something new already, and we're not even 5 minutes in.
Nice.
Dang. Okay. Okay. So let's talk about the problem a little bit more in the kidney space before talking about these specific publications as to why you're so interested in researching about them. Because there's a lot of math. I saw some of your
Yeah,
papers. There's a lot of math.
yeah.
Like it's in a
Oh man,
completely different language.
totally.
So about the problem space, specifically about
Yeah.
kidneys, what are some stats you can share with me that comes to mind that's kind of important to you?
Yeah, absolutely. So, firstly, so thinking globally, okay, kidney disease affects 10% of the population on the planet. So that's almost a billion people. And that is so if we're thinking about the importance of kidney disease, it not long ago, so maybe 10, 20 years ago, kidney disease wasn't even in the top 10 killers. And now it is in the top 5. So even
Oh,
so in this past period of time, there's been a rampant increase in kidney disease. And we're not really surprised by that because the most common causes of kidney disease are high blood pressure, diabetes, these chronic conditions that are increasing, right? And we have an aging population. But in the end, that's still a billion people. So something needs to be done for these billion people on the planet. And the way that, and I think, part of the reason why kidney disease has spread as much as it has is because there aren't that many drugs that are actually designed to treat kidney disease. These days there have now recently, like in the past 5 years, there's been an explosion of interest in treating kidney disease, but the vast majority of technology that focuses on the kidney is focused on the kidney after it fails. So these are patients whose kidney have already failed. That patient is going to go on dialysis, and dialysis will keep that patient alive for up to roughly 15 years at the very maximum. There are certainly people that live longer, but dialysis, your days are numbered on dialysis. The other option is
wow.
transplant, right? And
Okay.
that is extremely exciting, extremely interesting, amazing new science. I know wonderful, wonderful people that are doing amazing work in the transplant space. But again, that is only for people whose kidneys have failed. So if you look at, the billion people, and just for the record, it's 850 million.
Okay.
So it will be billion probably in the next 5 years, 5 to 10 years, it'll be a billion people. So looking at that billion of people, only a tiny, tiny fraction of those people. And so for example, in the US, okay, there are 37 million people that have kidney disease. Only less than 1 million people have kidney failure. So a very, very small fraction of that overall number are people that have kidney failure. They're on dialysis, they had a transplant, or they're on the transplant wait list. All the other people, there are practically no drugs for them until only very recently when a few drugs have come out for kidney disease. So really, there's a huge number of people that have no access to treatment for their disease. And this needs to be dealt with. Like there's no way that we can just allow this disease to continue. And so from my standpoint, of course I'm again, I respect dialysis, I respect transplant, I respect all the people that do all the amazing work. I'm more interested in what about these other 36 million people when less than a million people are on dialysis. And so that's what I'm mainly interested in, is how can we prevent the kidney from failing, as opposed to let's let the kidney fail and then treat the patient. In my mind, that just sounds kind of backwards to me.
Yeah. Yeah. So like all these people, they know that their kidneys aren't doing so great, and there's nothing that they can do right now. There's like literally nothing, and they just have to
So
wait until this doomsday situation for them, and
a lot
then
of the
maybe they'll
time,
get help.
yeah, so a lot of the time they don't even know they have it. And
Oh
so
shit.
what most people are saying now is that 9 out of 10 people that have kidney disease don't know they have it, that it is
Oh.
flying under the radar. And so if they, so one, they need to get screened in order to know that they have the disease. Then two, once they have the disease, the only thing they have access to most of the time is diet. Like, they exercise more, they have a better diet. They try to reduce their protein intake, they try to reduce their salt intake, increase their potassium intake, and that's about it. That's about all they can do. And of course, like, National Kidney Foundation, all of these organizations have all kinds of amazing information on how you can manage your kidney disease, but in the end, it is up to you and your lifestyle. It's not a treatment. There are some drugs, again, that are coming out primarily focused on rare kidney diseases, maybe genetic kidney diseases. So, but for the vast majority of people with kidney disease, it comes from diabetes. It comes from having high blood pressure for a long time. It comes from having heart failure. And there's very little options for these patients, of which there are millions in this country alone.
Holy crap. Okay,
Yeah,
so
seriously, you
holy
said it.
crap.
Yeah,
Okay, so if just for clarifying reasoning,
yeah.
so if you want to get screened, is this something that you can just go to your family doctor, or is this something that's like a specialist, like you have to ask for a special kind of specific screening that takes a long time, that only like there's one equipment available? Like, is it that inaccessible to get the screening, or is the screening pretty accessible?
The screening is pretty accessible.
Great. Okay.
if you do a basic metabolic panel, it will give you serum creatinine, which is now the standard method by which you can diagnose kidney disease. So if your serum creatinine goes high, it basically just means that your kidney is not filtering out the creatinine and it's not going anywhere. So if you have higher creatinine in your serum, in your blood, then that means your kidney is at risk of being in disease. So the thing is, though, I will say that your kidney can still have a problem and it won't show up on that test. So another layer to this complexity is that serum creatinine is not a very great marker. So it's not very sensitive. So if you have a problem with your kidney, it doesn't necessarily show up in serum creatinine. And that is another layer to this issue, is that you could get screened but then still not actually know whether or not you have kidney problems. If you have high serum creatinine, and if your estimated filtration rate, which is called EGFR, if your EGFR is low, in other words, you're filtering less, and it's very clear that you are not filtering as much as you should be, you surely have a problem with your kidney. But that doesn't necessarily mean that if you have a problem with your kidney, it will show up in serum creatinine. Yeah, I have a lot to say about this topic, but point
Yeah.
being, you can get screened, and it's not, and it isn't that hard. Like you can talk to your doctor, you can say, "I'm worried about kidney problems." They may screen you anyway. Like if you have heart problems, they may screen you for kidney disease to see if there's an issue. But again, it's pretty much always based on serum creatinine.
Got it. Okay. So, in the problem that you've been having a lot of fun, it seems like, the
Yeah,
last decade or so.
yeah,
I mean, you got your PhD in it,
yeah,
you did all these things. And so let's talk about the math for a second, because
yeah.
you mentioned how you said you're really focusing about inside the kidney.
Yeah,
And there's a lot of
right.
math involved. Can you just say a little bit more about what does that actually mean? Like, can you explain
Totally.
to me as if I'm like five
Yeah,
what that means? Okay.
totally. Yeah. So let's start with serum creatinine. And when you go into the doctor, okay? So when you go into the doctor will give you a number that's called EGFR, and that stands for estimated glomerular filtration rate. So the glomerulus is the filter in your kidney. Blood comes into your kidney and it gets filtered at the glomerulus. The glomerulus is a little ball of blood vessels. So the math involved is this, is that there's a pressure, right? If you have a filter and you have water on one side of the membrane, it will, and you push that water through, then the water will go through that membrane, right? That is the process of filtration. And in your kidney, that's how it works. You have a blood pressure at a certain level. Your blood goes into your kidney and it gets filtered. It's pushed through that filtration barrier with pressure. So that pressure changes. That pressure could be different if you have a certain disease. So if you have high blood pressure, that pressure goes up. You filter more through your kidney. So my research has been focused on predicting how in different disease states, in different conditions, let's say your blood pressure tanks, it goes way down. How does that impact the kidney? So using math, we can simulate all of those different scenarios. So
Ah,
we're looking at okay, under these conditions, how dangerous is this going to be for the kidney? So if you are diabetic, and if you have diabetic kidney disease, we can predict how bad that is going to be, which cells in the kidney are going to get hurt the most. And then from there, that means, okay, so now if we want to give a drug to the kidney, what drug do we give? We want to give the drug that helps those cells, not these cells, because these cells are fine. We want to deal with those cells, and that's the drug that we're going to give. So using math, the reason why we do this mathematically, right, is because otherwise, we're experimenting on a lot of animals. So we would have to run studies with many animals. And then, of course, comes the ethical quandary of, you're sacrificing all
yes.
these animals, and that's sad,
Yeah.
right? You
Yeah.
can go into the clinic and gather data in the clinic, but then you're running experiments on people. That's not easy to do, and it's not
No.
cheap, right? So in the end, math is completely without cost. The only cost it is my time. So using that, you run all these simulations, you predict what's going to happen. That is its own process that you can go through to then figure out, okay, now these are the hypotheses that we really want to test in an experiment in animals, in large animals, so like pigs, very important. Those kinds of experiments, very expensive. When you get into the clinic, it's like times 10 in terms of expense, both time and money. So
For the
being able,
pigs?
say that again?
For the pigs?
The pigs, well, yeah. So the pigs, I mean, if you go from mouse to pig, it's like 10 times more expensive. And then if you
Oh,
go from
okay.
pigs to humans, it's like 10 times more expensive than that.
That would make
So
sense.
yeah, it
Okay.
ends up being pretty
Sorry,
crazy.
I
So the
just
point is,
realized. Yeah, sorry, go ahead.
the math allows us to do that without those experiments. And
I
so
see.
it's part of the overall process. You can't solve these problems with math alone. But I would argue you can't solve these problems with experiments alone. You kind of need both,
Yeah.
and they work together.
Okay. So, sorry, I was just like, oh my gosh, I feel really privileged to be able to have in my science class in biology
Yeah.
to have dissected a pig. I
Oh,
really
yeah,
think
totally. Huge.
my junior high school
Yeah.
in Michigan.
Yeah.
By the way, Owen, I did a total mistake, and I accidentally cut off the tail of the pig. I wasn't looking, and so it was a group project.
Aw.
I know, and so I got deducted points, so I missed my A
Aw.
because of that accident. But
Aw,
yeah,
too
that
bad.
whole entire lab smelled terrible, and so
Yeah, that is a thing. You get used to it.
yeah.
Wow. After working with
Okay.
it. Yeah.
But the math of it, okay, so this is really interesting. I didn't realize going down that route, it also gives you an objective way. And when you're talking about these different simulations and scenarios, oh my God,
Yeah.
that's extremely exciting because
Yeah.
I know we'll probably touch on it later in the digital tools side, but I'm like
Yeah.
AI, right?
Sure.
Of like so many things.
Yep.
But how to like accelerate this because if there's a lot of these people who don't even know that one, they even have kidney disease, two, it seems like there's not much information about it, two, it's pretty limited because it's kind of like a niche in a way. And then three, it's like, even if they did know, what can they do if lifestyle is one thing, but at the same time, it's like, okay, I have to wait for like doomsday to happen, and then now what? And
Yeah.
so that's really kind of upsetting to hear. But at the same time, I think it's really amazing that you're tackling such an incredible challenge, right?
Thank
And
you.
I think what I'm hearing is like, the mathematician of you lives inside the kidney, and
Yeah.
then outside of the kidney, you're the scientist who's putting
Yeah.
together the thesis and hypothesis and
Right.
drawing up conclusion and trying to provide a better direction for people to conduct these experiments or
Yes.
creating these tools. Is that right? So it mitigates the risk of what
Right.
they're actually investing in to move forward, right? Because you're basically removing all of these outliers
Yeah.
that you can
So,
see. Is that right?
it's I mean, from the standpoint of I mean, if you're going to sit down and try to figure out how kidney disease progresses, that's really hard to do. Experimentally, that is, it's one difficult problem after another. And the big problem that you're dealing with is that you have 100 different hypotheses to test. So, okay, I think that it's this hormone that hurts the kidney. All right, that's one set of experiments that might take 6 months to do, and it's going to cost, let's say $100,000 to put together that experiment and do all that stuff. That's one hypothesis. So with math, we can test 1,000 hypotheses instantly. We can do it. We can look at all these different problems, all these different issues. And in the end, that, we can then identify individual hypotheses that are worth testing experimentally because we see, okay, the math suggests that at this blood pressure, you are going to see this result in a rat kidney, right? Well, then you test that hypothesis experimentally and you show that the math was right or the math was wrong. In either case,
Okay.
you learn more and you've moved farther and faster than if you just run at it, just bash your head against it experimentally, which, again, I have nothing but respect for experimentalists. I love people that work at the bench. I worked at the bench at Yale. It was amazing. I would like to make their job a little easier. That's kind of from my standpoint. That's how we can de-risk their experiments, de-risk the ultimate result, which hopefully is a drug for the kidney disease.
And you're also just like racing with time, right? And so like if there's only 15 years, I think I understand why you're saying about the doctor earlier. You mentioned
Yeah.
Gabby, right?
Yep.
He's not retired. He's the only one who's not retired because there's still a
Yeah.
lot to do.
Yeah.
Um,
Yeah.
and
There's
so
so
if you're
much to do.
Yeah, and if you're catching up with time, I mean, it sounds like what you're doing, you bring the math side is kind of accelerating the process so that you can figure out these scenarios. Okay, so
Yeah.
the math side, okay, so help me understand why, or help me understand what was the response of the papers that you did in like help me understand, like give me an example of one of the papers that you did. Let's talk about drug delivery. First of all, if you can contextually explain what kidney drug delivery is, because it's probably
Yeah.
not what people are thinking. This is not, you know, door dashing kidney drugs.
Totally.
It's like it's not like
Yeah, absolutely. Yeah, when I say drug delivery, yeah, a lot of the time people are like, "What now? What do you do? You do that at Yale? Interesting." Yeah, so drug delivery is so as a basic definition, when somebody says drug delivery, they're talking about where the drug goes in the body and how fast it gets there and how long it stays there. So a good example, if you have a vaccine in your body, you want that vaccine to get to as many cells in your body as possible, so that the vaccine can reprogram the cells so that they will respond with an immune response to the virus that comes in. The key thing is that maybe this vaccine is small enough that it gets filtered by your kidney. And so as a result, the vaccine collects in your kidney and it binds there. Now,
it
maybe
collects.
if it collects in your kidney, because some drugs are bound to bind within your kidney. They get filtered in your kidney, but they don't end up coming out as much in the urine. And so then it stays in your kidney. And that's when you can then get toxicity in your kidney. So drug delivery, the point of drug delivery is to get the drug to the right cells and not to the wrong ones. Sometimes you want the drug to stay in your body for a period of time. So, nobody wants to get an injection every day, right? What if you could get an injection once every 6 months? Well, the way you do that is that you have to package that drug in the right way so that it's maintained in your body for 6 months. That is the discipline of drug delivery. And so that's what I worked on
Okay.
at Yale, looking at specifically how to deliver drugs to the kidney, which is a big problem, a big challenge.
Okay, so you did this at Yale
Yeah.
and the laboratory, and help me understand contextually the environment of like where you did this research.
Yeah, totally. So in this case, so this was a really good example of what we were just talking about with hypothesis generation with math. So when I first got to Yale, I was working with Greg Tejan and Mark Salzman. Greg Tejan, so at that time was an assistant professor at Yale. He's now the CEO of Revalia Bio, which is a startup. And so just speaking about Revalia for a second. So Revalia, what they do now is they perfuse human organs. So they get human organs from typically car accidents. So when somebody dies, their organs, if they are an organ donor, that organ goes up for potential transplantation. If it is found ineligible for transplant, of which there are 1,000 reasons why they won't transplant an organ, because you never want to put an organ into somebody unless you know it's going to benefit them. So as a result, there's a lot of organs that are not eligible for transplant. Those organs can then be used for research if the patient signs off. Like there's an entire process by which that occurs. The point is, no matter what, human organ experiments are super expensive. So Revalia is a a startup system. They just received some investment to perfuse organs at scale, and perfusing means pump blood through these organs at normothermic temperatures. So essentially, what that means is that you wake the organ up. So the organ shows up on a box of ice. I know it's like Frankenstein, totally. Shows up in a
That's
box of
literally
ice.
what came up to
I
mind.
know. You plug the organ in to a pump and it pumps
Okay.
blood through. And so if it's a kidney, this kidney will produce urine. So
Oh shit. Okay,
the
I
person is
understand now.
dead, but the kidney is still alive, right?
Wow.
Okay. And so why this matters, right?
Yeah.
From an application standpoint is transplant. So the future of transplant will involve perfusion. So while the organ is outside of the body, so it comes out of the donor, it goes on a pump and then has blood pumped through it to keep it alive, then it goes into the recipient. So what's cool about this specific system is that now you can give that organ drugs. So when it's outside of the body, it's on its own. You can do anything you want to it before you put it into somebody else. Now imagine for a second.
Wow.
Yeah, I
Hold
know.
on. Wait, hold on. Owen,
Crazy.
you're saying you can beef up this organ basically and have it like have superpowers and then
Hmm.
you install the organ
Yep.
and it's supposed to work with everything else.
That is the idea. Yep.
Okay, sorry. Okay, just
Because now what's done, right, is that an
uh-huh.
organ is transported on ice. So
Yeah.
and then you have this cold organ that you put into somebody's body. So the way that they're doing it now, and so companies like Organox, which was recently acquired by Teramo, they do this for the liver, which is a different ball game, but that currently liver transplant involves in a lot of so in the UK practically every liver transplant involves perfusion and then they put it into somebody. And so that moment when you have the organ outside the body and you can do anything you want to it, you can give it drugs. So in this lab at Yale, so as a collaboration between Mark Salzman and Greg Tejan. So again, now Greg is at his startup. They do this commercially. They perfuse
Okay.
human organs and they essentially run virtual clinical trials with these organs. So they can run a clinical trial without people. They just have
A
organs. They
virtual
run.
clinical trial.
Yeah. And I mean it's actually honestly it's not even virtual. It is a clinical trial. Like you can run a clinical trial in organs. So this is a thing now that people are getting into. It's complicated from a lot of the regulatory perspective. There's a lot of aspects to it that are very complicated, but there are currently clinical trials being run on organs outside the body.
That is wild.
Wild. I know. It's
Okay,
crazy.
the only clinical trial experience I have is with Reata Pharmaceutical and
Yeah.
like those kind of drugs, like normal
Sure.
clinical trials.
Totally.
This is like extremely modern and advanced. Okay,
Yeah. So, and that's Revalia Bio.
wow.
So, just, shouting out, giving it to Revalia. There's other companies like Pebble. So Pebble Bio uses pig organs, but does the same thing. They perfuse organs and they give it novel therapies and then see how the organ responds. And so before Greg left to go to Revalia, he was at Yale, and Mark, my advisor at Yale, is a nanoparticle guy. So what Mark does, and what Mark has been doing for decades, is his lab creates packages for drugs, and these packages are, you know, smaller than a cell, but they're larger than a molecule. So they are actually pretty big containers that hold drug that then can be injected into circulation. They last longer in circulation than the drug alone. And the other key thing is that with these nanoparticles, you can do gene therapy. So you can have a genetic code inside that nanoparticle. The
Holy
nanoparticle
shit.
goes into the cell and it can reprogram the cell. So part of the
Okay.
reason why the COVID vaccine technology was so groundbreaking and controversial is not just because it was developed quickly, it's because it was a completely new way of making a vaccine. What they did was they put a genetic code. So the COVID vaccine is gene therapy. They have a genetic code inside a nanoparticle. That's how the COVID vaccine works. It's a nanoparticle that goes inside the blood and then eventually reprograms cells to recognize COVID. The same idea was what we wanted to do at Yale. So what we wanted to do was we wanted to have nanoparticles that we put in to a kidney outside the body, and those nanoparticles would deliver a gene of some kind that would change the way that kidney would respond to immune cells. What that essentially would do, if that worked, right, is that would be the holy grail of transplantation. Because what you then can do is you can give me a kidney. It doesn't matter what your blood type is, we can change that about this kidney.
Oh
It
my
doesn't
gosh.
matter what HLA allele you have, we can change that. That's the idea. Now, we're far from that,
Wow.
absolutely, but that is the idea. And this technology, I mean, the thing is, we can hit a lot of cells with these nanoparticles. Like, you can get there.
Wait,
You
hold
can
on. Owen,
get,
this is why you're saying the kidney being outside and like injecting that, doing whatever you want with it before it's being installed. That would be a thing because if it doesn't matter about the blood type, then you guys can change whatever that is. Whoa, that's cool.
yeah. And I know, I mean,
Oh,
for
wow.
example, I mean, I know the wonderful people at Cambridge, University of Cambridge, that they have successfully changed the blood type of an organ. They've made a kidney, type B kidney, become a type O kidney. They've done that.
Wow.
So, and there are
Okay.
groups in Toronto doing that in the lung. But
Science is so cool.
blood type is simply one of the
Hmm.
many, many factors. Like,
I see.
it's crazy. The
Okay,
point
this is a
being,
playground. Wow.
yeah. So these experiments are kind of like a playground, right? Right. Like you have access to a human organ. Wow. First of all, whoa. Like that's incredible.
Yeah.
It brings a tear to your eye. It's funny because it sounds so gruesome, but that's a part of a person. I mean, wow. You know, and you get to honor that person. Wow.
Yeah.
That's amazing. But the point is that no matter what you do, so these experiments at Yale, there were six kidneys, three pairs of kidneys. That took about 4 years to do. So that's a long time. That's how long I was in my postdoc total at Yale. It's also thousands upon thousands of dollars. So every experiment, like even if you're doing it cheaply, it is
Mhm.
very expensive. So this is when math comes in. I can try all kinds of different systems. So what are some things I can change? I can change all things about the nanoparticle. I can make the nanoparticle bigger, smaller. I can change the material that I use. So sometimes we have polymeric nanoparticles, so biodegradable plastics. Sometimes we have fat nanoparticles that are made of lipids. So there's lots of different options. You can change the pressure that you're perfusing this kidney at. All of this can be done on the computer without an experiment. Those experiments cost thousands of dollars. So I would like to set up and figure out what experiment has the highest value that we can then ultimately develop a therapy that will actually come out. And we can do that in 3 years instead of 10.
Yeah, that would be ideal, Owen.
That's the idea.
Yeah, and bring that number down of kidney disease, right?
Yeah.
Wait, okay, so what you're saying, let me I just want to make sure if I am understanding this correctly. So
Oh.
as you have these different controls, right? So these controls, because every human is different, everyone has every human is unique, literally
Yes.
rare.
Yep.
And so by using math and creating these like systems in a way, you can then change the dial of like, okay, this one has a lot, or this doesn't, or it should be in this way, da 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 da. So then you have these unique combinations that you can get close to, and that allows you to create that simulation to get to the closest, most accurate representation of the scenario that you're
Yes.
dealing with. So then at that point, you're just really the experiment is talking about the circumstances and the environment that it needs to play out, and then it's all these other variables like, and also is there factors with the heart, and also
Yeah, so that's the
okay.
thing, right? Is that in an experiment, that's when you're dealing with the nitty gritty details. Like, okay, you have a nanoparticle formulation. Did you spin the vial before you put it in to the system? Before you injected it into the kidney? Did how large exactly was that batch of nanoparticles? As like, if I have one vial versus another vial, if the experimentalist made a mistake, if the kidney was larger than expected, and this is what, because again, that kidney shows up and you're working with it, and then that might be at 5 a.m. and maybe there was some small source of error in the experiment. That's never going to be captured with math. You're not going to capture that with math. That's not the point of math. The point of math is to figure out all the ways in which you can waste your time so that you don't waste your time. And that experiment, it's just that experiment that you have to absolutely show that, yes, this drug does work. And we already tried all these other scenarios mathematically, and we found this is the best choice we can make. It's at this pressure, this flow, etc.
That's a beautiful and refreshing way to talk about math, Owen.
Yeah.
Oh my gosh. I love that so
Yeah, I do
much.
too. Yeah, totally. Yeah.
Okay, so let's talk about these papers real
Yeah.
quick. So help me understand what kind of papers did you do? So you were first author and also a second author on a whole bunch of papers.
Yeah.
What are your favorites?
My favorites. Well, really, yeah, my favorite. So we published a paper. It was my first author paper. And that was a big favorite of mine because it was the hardest. It was one of the hardest papers to publish because I had this idea. And I wrote some code. I did some experiments. I wrote up a paper. We submitted it. Rejected. It was rejected with flying colors. Like actually the editor of the journal said, if you want different reviewers, we can get different reviewers. Because the reviewers were so incredibly not into this paper. And they let me know.
What a
There
peer
were-
review, Owen.
Yeah. Science is supposed to be impersonal. It was personal. It got personal. And those reviewers had some of the best feedback. Very scathing, very difficult, but actually very correct. Their feedback was very correct. And that in reality, we just didn't do a good job. And there's a lot of reasons why. So I went back and redid everything, 100% everything, and resubmitted, and we made it into a journal. And so for me, that was the big one, right? And I think I matured. I mean, I think really that's when I matured as a scientist, was like, oh, it doesn't matter how much you want this to work. If it doesn't work, it doesn't work. Like, that's it. If the reviewers don't like it, it's not going to get published. You have to do something that makes sense to readers that really do know what they were talking about. These reviewers were fantastic and a little scathing. So that was a great paper, and really, and that had nothing to do with drug delivery. That was focused on the kidney, blood flow in the kidney, looking at different disease states, looking at different blood flows, and yeah, it was great. Fast forward to now, and I'm actually still using that exact same model. I'm using that model, but I'm looking at nanoparticles in that system. And so now I'm using that model not to look at the kidney and like disease states in the kidney, but to think about how nanoparticles get distributed in the kidney, which is that's much less about physiology. That's much more kind of about nanoparticles and nanoparticle physics.
Wait, so is this why you're mentioning about how, rather than waiting for, or like the example how you gave about like if people were getting injections every single day, that would kind of suck.
Yeah.
So how do we do it like that? They get an injection like once every 6 months, and then that's where the nanoparticle thing is. So then that's where, with Mark Saltzman, you guys were able to really work on the nanoparticle aspect of it. Okay, I understand. I think I'm starting to understand where, okay, I
Totally.
get it. Okay, so going into drug delivery, okay,
Yeah.
got it. Sorry, I'm like
Right. No
learning
problem.
so much right now. Okay.
Yeah, but I mean, so in this case though, so actually specifically, what we're doing there. So you have a nanoparticle. The thing about nanoparticles is you can do things to nanoparticles. So if I have a drug, or if I okay, so if I have a nucleic acid genetic code, right? I can modify that molecule directly. That's one option. So if I want that molecule to go into a cell and maybe a particular cell type, I can modify that molecule. The other way I can do this is I can take that molecule and I can put it inside a particle and then modify the particle. So in this case, what we did was we put in these studies, we didn't have nucleic acids, but the idea is that you put it in a nanoparticle and you put antibodies on the nanoparticle. The antibodies stick to certain cells, and those certain cells will take them up. So the reason for the nanoparticle, in the case when you have a transplant organ that's getting perfused and then it's going to go into somebody, that those nanoparticles hold genetic code, but they can also be targeted to particular cells. And so that's what we were looking at in this study. And
That's
from
cool.
from my end, so now talking about like first author versus second author, right? I'm not first author on these experiments. These experiments took 30 people to do. Like
Wow.
like wild. Like it's not, this is not something that I'm qualified to be first author on. But so I helped with those experiments. So I'm on that paper. That's great. Published in Nature Communications, very exciting. Mark Salzman, Greg Tejan, Jordan Pober, who's a really big, very well-known immunologist, very exciting. And I love to be part of it. It's one of my favorite parts at Yale was being part of that project. But the first author paper that hasn't come out yet, but will, I'll give you a sneak peek. It's focused on the math side of that. So taking the data from those experiments and then doing it mathematically. The idea then being that, hey, you want to run another set of experiments, you want to do another study, here's what you should look at. Nanoparticle size doesn't matter. Nanoparticle charge doesn't matter. This is what matters. This is the variable that you should test when you spend $100,000 to run those experiments. $100,000 that scientists don't have, especially these days. But that's another
Yeah. So
conversation.
you're taking out the guesswork.
That's the goal.
Okay. That's really freaking cool. Okay,
Thanks.
hold on.
Thanks.
So, wow. Okay. Okay, so I'm just like, okay, I can understand now the math side of you, and then there's the science part of you, and then help me understand the engineer side of you. You were saying that you were writing code, and then you did the paper part. Is this part of your process?
Right. So, yeah. So, and this is, I think this is also like a good kind of question that is on a lot of people's minds right now, which is what is an engineering in academia, right? So, like,
Okay.
because
That's
engineering,
yeah.
like, so engineering, like biomedical engineering,
Yeah.
is a
Software
very,
engineering too, specific.
yeah, so biomedical specifically is varied. So people say they're a biomedical engineer. That could mean a lot of different things. That could mean one, that can mean they're building implants for your hip replacement. Straight up. That's what they do. They're a biomedical engineer, so they make hip replacements. They make robotic hands. They make robotic arms. They do the big stuff. Then there's biomedical engineers that are programming cells to seek out cancer and kill that cancer. That's also a biomedical engineer. Those two skill sets are completely different. Like
Right.
there's no overlap, right? They're completely different skill sets, but they're called a biomedical engineer. So in my case, I am a coder, right? That's what I do. I do math every day. I'm a biomedical engineer. And I think a big thing, like when people talk about the difference between science and engineering, I think a big component of engineering is that you produce something. So Mark Salzman is a biomedical engineer. He was the inaugural chair of the biomedical engineering department at Yale. What Mark does is he makes drug delivery mechanisms. So a nanoparticle is an example. An implant is another example. That's what Mark makes. And that's what makes him a biomedical engineer. You can find something out. You can learn something. That makes you a scientist. But if you make something, that's an engineer. So I make mathematical models which are represented as code of the kidney. I find out things from running experiments with that code. So I run the code, thinking, oh, what if this kidney is, big, and what if the kidney is small? But those experiments, that's science. But the actual production, the model that I'm using that's coded, that's online, that you can find, that is my engineering. And so in, going forward in my career, I would be very excited to engineer new ways of finding drugs for kidney disease, preventing kidney injury, preventing kidney disease from progressing. And all of that ends up being a product that is a code. And maybe that code shows up as software that is sold, right? And I make software that I sell from a company. I'm not there yet. But that is my engineering. Ultimately, is software that comes out.
Amazing. Wow.
Thanks.
Thank you so much because it's articulating the difference. I think, people who are in grad school, people who are like, oh my gosh, how do I have a career like you do?
Hmm.
It's incredible because you're intersecting all these different disciplines, but your strength is it's still underlying this umbrella of this core mission about eradicating kidney disease.
Yeah,
And
totally.
I think when zooming out to that, it's like these papers, these research, the peer reviews is so incredibly important, like how you're saying about the critique and the
Totally.
feedback. But
Huge.
what you're saying, how like you're so humble about it, and I'm like, oh my gosh, thanks for being so like
Oh,
open about
totally.
it wasn't really a failure. You learned so
Oh yeah.
much in that
Oh yeah.
process,
Big
but
time.
then the standards and the integrity of science allows for the incredible amount of feedback, the objective feedback by different perspective for you to look at that each advisory basically and be like, okay, how can we do this making better? And then resubmitting that. That's incredible. Oh my
It
God, Owen.
it's a normal process.
Wow.
The first time I did that, I was like, "Oh, I'm quitting science." Like that was, very much a real response. I was like, "Oh, these people that are so knowledgeable, they hate me and they hate my work and I feel so terrible and I'm gonna drop out." I thought about that for maybe 24 hours. The thing though is that that has consistently been my best work, is when people tell me, I don't like this, I think you're doing it wrong, and then I redo it. And in the end, like, and I think, I mean, in the world of drug development, you're looking for the killer, right? And by that, I mean, you're looking for the killer experiment, the experiment that you can do that will kill a drug, that says, actually, this drug is not going to work. If you know something isn't going to work, that's gold, gold,
Yeah.
because you aren't wasting your time on something that won't work. And it's amazing how easy it is to believe your own BS. It really is amazing. And like I am so susceptible to that. And I am really just trying to not believe, just try to like just look at things objectively. It's amazing how hard it is to do that. But that's the job. That's what a scientist does.
I feel like you should speak at some startups
Yeah.
because that
Yeah.
right there.
Maybe, we don't need to go there. But yeah.
That is incredible. No, it's a great way to be thinking about. It's a wow, that's very humble. Okay, so back to the papers, because one paper did catch my eye, and I can't not talk about it, Owen.
Sure.
You know exactly what I'm gonna bring up.
I know exactly.
I mean, it's the one that I was able to like read and comprehend the most.
Totally.
What the hell? Why do you have a sperm paper?
Yeah,
Owen?
totally.
What
So-
does that have to do with kidneys?
Yeah. So okay. And then yeah, this is I started as an undergrad. I started doing research on sperm. Okay, and the reason was because of at Tulane, so Tulane University, where I did my undergrad and my PhD, there are superstars in mathematical modeling of microorganism swimming. So I know it sounds crazy, but actually follow me here. So Ricardo and Lisa, so Ricardo Cortez and Lisa Fauci, these are, rock stars in the world of modeling the fluid dynamics of how tiny organisms swim. So my first research project as an undergrad was looking at multiple sperm and how they swim together. Because in biology, and this is kind of a wild thing, but in some species, the sperm of a male will team up to fertilize the egg. We talk about it like it's always a race, but it isn't always a race. It actually, in some species, it's like a race between teams. So we were looking at the math of how do fluids impact that. So these sperm are together in the reproductive tract, and they're swimming together, and they influence each other through the fluid. Now, firstly, let's think about this from the drug delivery standpoint. Okay, this is why this matters. Okay, sperm. So why, Owen, why would you possibly be interested in sperm swimming? What does that matter, right? Well, guess what? If you're trying to get a drug to an egg, an ovum, right? So let's say, for example, person with the egg, mommy, she has a genetic condition, and we want to alter her egg so that the baby won't have that genetic condition. You know how you can do that? You can give a drug to a sperm and then let the sperm find the egg.
To a sperm?
Sure, a set of sperm. So you have some sperm, you give them drugs, the sperm will find the egg. That's their job, right? And they will deliver it to the egg. That's drug delivery. So that probably is the best way to get a drug to an egg, because the sperm will always find the egg. Now, so hence why we're interested in sperm. But the really interesting story is what's the relationship between sperm and kidneys? Because actually there is a relationship, and that relationship is very interesting. Much of the sperm research is funded by the National Institute of Diabetes, Digestive, and Kidney Diseases. And the reason for that is because polycystic kidney disease. So polycystic kidney disease is the most common genetic form of kidney disease. So these are patients that genetically are predisposed to their kidneys growing to the size of footballs. And so actually their kidneys get so big that they have back problems. These people, these patients, and I've met some of these patients, they often will lose kidney function in their 50s. And so if you think about that, and then dialysis is your only option, you're living till about 60. And actually, what I've also come to learn is that these are the lucky patients. That actually many patients, yeah, that many patients with polycystic kidney disease, they have aneurysms and just die, and we don't know why. There's all kinds of like just wild problems that like are very unexpected. And guess what? We know so much about the genes that cause polycystic kidney disease. And what we know is that these genes control cilia. So specifically the tail on cells in your kidney. Now, the cell that we know best that has a tail is sperm,
Got
right?
it. Okay.
Right.
I'm tracking.
So
Yeah.
if you look at somebody's sperm, and this is an open research question, like, hey, if somebody watching this wants to look at this, please, by all means. If you look at the sperm of someone with polycystic kidney disease, do those sperms swim differently? Is basically the question. So now you've gone from sperm to kidney disease. And guess what? I mean, it was a complete coincidence that I happened to start in sperm and then go to kidney disease. And Ricardo Cortez, again, sperm expert at Tulane, he was my
Oh,
PhD
he's a
co-advisor
sperm
on the kidney.
expert.
Yeah, he's a sperm expert,
Okay, got it.
you know?
Not every day you hear someone.
Not every day you hear something like that. It's not every day you hear of somebody being a kidney expert either. So there you go.
That's true.
You know? But you know, in fact, if
Wow.
somebody says that he's a kidney expert, I think you'd probably expect that I would be an expert on kids' knees, like I'm an orthopedic guy or something. Pediatric orthopedics. Anyway, point is, yeah, so that was my first paper, and it's a good paper. I like that paper. It was great. You know, we looked at possum sperm. We were thinking about all this stuff, and I'm lucky now to be in the kidney world. I get fewer laughs. I get fewer laughs now that I'm in the kidney space, even though I look at pee. That's like part of my job. So anyway,
Oh, okay.
you know,
Yep.
there
Yeah,
you go.
that makes sense. Yep. Renal stuff too, right? All the things.
Yeah.
Right. Okay. So, okay, no, thanks for entertaining my question about why sperms and kidneys
Totally.
have a collaboration there. And well, Owen
Yeah.
did it.
Yeah, totally. If
Check.
I had funding for that project, I would know exactly who to work with. And yeah, but
Who would that? V.
there's Tulane people, but there's some other people I know
Got
that
it.
would be
Got it.
interested in that problem. Yeah.
So, okay. So those are about your papers. Now, tell me more about Yale. What did Yale teach you in a way? Because in your postdoc, you know, you were in the academia world for quite some time, and so as you are now at Novartis, and so that has been a switch. So reflecting back, because it's so fresh, what has Yale, the postdoc, what's your takeaway? If there was a main takeaway.
Totally. So, first thing I would say about my experience at Yale was that I feel like I matured more as a scientist in that postdoc than ever before. It was such a huge learning experience. It was eye-opening. I learned so much about not just about kidney disease, not just about drug delivery as topics, but more about what scientists on the bench go through. I learned about that anxiety. I learned about how how you've already rehearsed the experiment 1,000 times before you do it. That rehearsal is happening in your head. Some of the best experimentalists I know, they already know what the problems are. They already know what the problems are going to be before they encounter them. I mean, really, there were amazing people at Yale. And I cannot speak highly enough about my advisors, about the people I worked with. Wonderful. And Mark Salzman is an angel on earth. He's such a great guy. Okay, so all that said, right? You know, I think the key thing that differentiates Yale and Harvard, these high prestige institutions, as compared to Tulane, which Tulane, great place, awesome stuff going on, not quite as prestigious as Yale. They don't have as much money. They don't have as much resources. So as a scientist, okay, you want to publish in Nature, you're going to need a lot of resources to do that. So by resources, I mean microscopes. By resources, I mean connections to people that have rare animal models. Like, oh, I have a mouse model of this disease. Those are not easy to find. You can be working with run-of-the-mill stuff, but if you want something really special, so like immunodeficient mice, you want to do human organ experiments, like none of that is going to be possible if you don't have a lot of resources. And Yale has a lot of resources, and that is really awesome. But here is the double-edged sword, is that if you have a lot of resources and you don't do well, it's your fault. It's nobody else's fault. It's your fault. And that was always, I mean, it's funny because in fact, that's always the case. That if you don't do well, it's, you can't blame it on somebody else. You have to recognize that the fault lies with you. But if you don't have the resources, it's a very different situation because now, oh, well, I didn't have this super crazy microscope, so this is as much as I could do. Okay, there you go. But guess what? At Yale, you do have that crazy microscope. You do. You have all the tools you could possibly want, and you still didn't get it done. Tisk tisk. And that is pressure. There's a lot of pressure, and that is scary. And it starts making you think, what's the limit of my mental capacity? Maybe one of the first times I had to ask that question, can I ask the right question? Like, am I smart enough to do that? It's funny because until then, I always thought, well, I know what question to ask. And it's simply a matter of not having enough resources to answer it. But now I do have enough resources. And now suddenly the questions you ask, how much time you waste, suddenly becomes so important. When as before, if when I didn't have resources, it was fine for me to waste time. That's okay. And I was in a PhD. You waste a lot of time in a PhD. It's part of the job. It's part of the job. As a scientist, I think you could ask any scientist and they would all tell you that you waste a lot of time. I'm pretty sure that's a very big component of this job. The point, though, is that, and being okay with that, right? But at Yale, it's hard to be okay with that because it's legitimately your personal failing. That's the way it can feel. And it's not your personal failing that causes you to not do well. But it's really easy to go there. And so for me, there were a lot of challenges in that regard. It was also really challenging because this was my first time working on the bench. I had done some experiments in my PhD, but really very minimal. And when I got to my postdoc, I was on the bench every day. I was making nanoparticles. I was learning how to put antibodies on nanoparticles. I was learning how to put those nanoparticles on cells in a cell culture hood. These were things that I didn't know. So I actually really started from zero. There were certainly undergrads that learned faster than I did. And it was very humbling. So like if you talk about like humility, I mean that's the way you learn humility is that you learn from some like incredible genius people. Like the people I worked with at Yale, some of the smartest people I've ever met in my life. Also surprisingly anxious, very anxious people as well. Right, but you can work with all of these people, and that challenge, like I was never not challenged. And that's great. It's also really humbling and really hard to do that as a postdoc when you're supposed to know what you're talking about. That's like there's this pressure, right? As a postdoc is that you're supposed to know what you're doing. Like even though you don't. Like as a researcher you never know what you're doing, but like as a postdoc you're like supposed to know, and I really didn't. I was just a baby boy. And so yeah, it was an amazing experience. Like, I totally understand why people hold Yale in such high regard. I get it for sure, and it's awesome, but super challenging. So it was really a hard time in my life. It was a hard time, and I would not have it any other way.
Gosh, so that really it would make or break a person, basically, is what you're saying.
Yeah.
And you
Yes.
not just survived, but you thrived in it.
Yeah.
And
I made some
wow,
mistakes. If I could go back and do it differently, there would be a couple things I would do differently, for sure. But it certainly didn't break me. It made me into the person I am now. And for that, I am so very grateful. But I did see it break some people. It's
yeah.
crazy, It's a lot of pressure.
So, so when it comes to when you're talking about like the standards that are held, right, and the
Yeah.
competition, resourcefulness, and the mental capacity of like, okay, I can't give an excuse basically that I didn't
Right.
have the fancy microscope.
Right.
I can't give the excuse of XYZ that I would normally do.
Hmm.
And I think that's a great way to put it, Owen, in a way of like, why are you there in the first place? What are you researching? And
Right.
do you have the skills and knowledge and guidance and the camaraderie to be able to move the needle on something that's incredibly challenging?
Yeah.
And so it's kind of like going through that, a little bit of getting your head out of your ass and
Yeah.
trying to really push every single boundary possible. That's what it seems like. And it's like
Yeah.
you're like literally breaking through.
Yeah.
But in the guidance in it's like almost like you were in a really fancy trampoline
Yes.
that
Totally.
yeah, it like really really fancy one,
Yeah.
and having all the guardrails, right, so
Right.
that if you accidentally veer off, you're gonna
Right.
come back in basically, and you're insured in that way. But it's almost necessary because of how much access that you would have to these very like almost like IP like
Yeah.
situations, right? You're saying these very
Literally. Yeah.
specific models.
Literally IP. Yeah.
Yeah, and
Right.
so it's really interesting how you're putting it in this way. I'm like, gosh,
Yeah.
this is like no different to like startup situations and
Sure.
technology.
Totally. Yeah. And
So
actually,
let's,
and
yeah.
also working with Greg, right? So Greg left to go to a startup. Working with Greg was like being in a startup.
Wow.
It had that same energy, which I loved. I was super excited about it. And I stayed at Yale because I wanted to get more papers out and I wanted to do more stuff. Again, awesome that Greg went to Revalia and that energy is awesome. And there are so like it's amazing the energy at Yale because people it's much more about what do I have the bandwidth and the time and the energy for. It's not about, well, what can we do? It's like you can do so many things. Like if you're at Yale, you can do so many different things. You can talk to the experts on everything. You can ask them, oh my God, amazing, right? But we only have one body, and in my case I'm lucky to have two hands, and that's it. That's all you have access to, and you only have one brain, and you can only think so much and only ask so many questions. So it, you know, it's a wild environment for sure. And I was very
Yeah.
lucky to get a piece of it, and I thank Mark for that. He was such a great advisor.
Yeah, and you, as you were telling about Greg's story too, what came to mind, oh, and I'm like thinking like, wow, he really is a really smart cookie,
Yeah,
because because as you're explaining about kidney transplant,
yeah,
how logistically it happened as to when in the like almost journey of a patient,
yeah,
right? What would they need? And he's
mm,
like right in there in
yeah,
at the time when they would need that specific organ, and then also positioning himself to be able to be ready for being able to insert whatever superpowers to then
sure.
transfer it. But in the midst of all that, I'm like, man, the real winner in this, I think, is in the distribution logistics. Whoever is refrigerating
Yeah,
it all,
right,
they're
right.
I'm like
Yep.
thinking, man,
Oh yeah,
they're the smart
they're
ones.
moving, and so the company
Wow.
that
Woo.
needs to be shouted out is 34 Lives. So when we're talking about transplant organs, transplant kidneys specifically, and treating transplant kidneys before they go into the patient, I think the people that have the best possible chance at doing that and doing that very well is a company called 34 Lives, and it's run by Chris Janes, who I've known of Chris Janes for years, years and years. He's been in the kidney world since before 34 Lives was started. But now what 34 Lives do, what that company does is they perfuse kidneys and get them into patients. That's what they're doing.
Oh, they do the whole
And
end-to-end cycle.
yeah, so they are
Okay.
the ones I think that are going to be best positioned to treat these kidneys before they go into the recipient. Because that's not like
Interesting.
you can't do that right now. Like that's not
Yeah.
like the FDA doesn't approve of new drugs to be done in that way, right? So, but 34 Lives, like they got my vote. Like because they're great. They're awesome. And they've done so much good work. They have amazing people. They know Greg. This isn't a very big world, right? So, you know,
They're
forget
probably
about it.
hanging out right now.
But and they're just such nice people. Also, Henri, and
Oh,
Chris are just lovely guys, lovely people. The point is, so if you want to like follow that, and if somebody watching this wants to follow that, 34 Lives. That's the
got
company
it.
that you want to watch because they're I think they're going to do it. I mean I'm pretty I believe in them. Like they're doing good
Yeah.
stuff.
Wow. Wow. That is a convicted with much conviction of that
Conviction.
bet. Yeah. Okay. So going into digital tools.
Yeah, digital tools.
So biomedical industry, like just biotech in general.
Yeah,
Biotech is huge. Like,
big,
oh my
huge.
gosh, right?
Right.
There's a lot going on there. It is an incredible place. It's kind of the wild, wild west.
Hmm.
At the same time, it's a growth area.
Right.
You have to go really fast.
Yep.
At the same time, there's some pretty heavy things that you have to be regulated on, and there's a lot of compliance involved too because it's such
Yep.
real-time data that you're dealing with.
Yep.
And so whether it is the actual drug delivery
Yeah.
of like
Right.
literal drug delivery to the
Right.
patient, right?
Right.
And distributed through to the doctors and hospitals and surgeons and whatever, of
Right.
those channels. But like how what are you so excited about from an innovation standpoint? What
Hmm.
is your perspective? Because you are a very interesting person who has this incredible lens from a math side, scientist side,
Yeah.
engineering side. So help me understand.
Yeah. So, okay, this is the deal when it comes to biotech in general and like what the role of digital will be. So, digital, which which encompasses AI, but AI is simply one form of digital tool that is being used, right? So AI is one way by which you can use digital tools to speed up the process of development of therapies and medical devices. So the game okay, is to speed up the process of developing new drugs, developing new devices. That's the game, okay? Because in the end, digital tools are not going to keep your heart beating. That's not going to happen, okay? So it's a physical drug that needs to be made. It's a physical device that needs to be made. So in general, so let's think about drugs, right? It takes the average drug about 12 years to go from development to marketed. And honestly, 12 years to me sounds pretty fast for how a lot of drugs go. A lot of drugs take a long time, right? The
Because they
reason
get
being
stuck
clinical
too in
trials.
FDA as well.
Yeah, so the FDA is and I have only good things to say about the people that work at the FDA. I love people that work at the FDA. They are hard to satisfy in terms of all the data that you need to show them in
Yeah.
order to progress to phase one clinical trials. But then you have three phases. So
Right.
right. Okay. So the like I was talking about with my models being used to de-risk experiments, digital tools, like this is the purpose. They're there to de-risk drug development. They're trying to make it happen faster. They're trying to, and the way that you make something happen faster is that you figure out what drugs won't work. You're looking for the killer. You're trying to kill as many drugs as you can. The ones that make it through that, those are the ones that are actually worth spending years to get them through the FDA. So digital tools can be used to at practically no cost. Like in comparison, so a typical drug costs about $2 billion to get to market, right? If you have like even the biggest souped up computer that looks at every possible like combination of amino acids that go together to make a protein that you might want to use to treat a disease, a fraction of that amount of money would be used. So ideally, you would be able to find all the drugs that won't work so that you can then test the drugs that should work based on the digital tools. So these digital tools are solving the model problem. So the model problem, or at least what I call the model problem, is the fact that the only way to know how a drug is going to act in a person is to put it in a person. But you can't do that because you might hurt the person, and that's not ethical. We can't do that in clinical trials. So instead, we have to figure out ways to predict how a drug is going to act in people. Animals are one way to do that, right? You can, use cells. There are certain animal models that might have human components, so they can be bred or, genetically manipulated to have certain proteins that are human proteins, not mouse proteins. There's all this stuff. Well, the same thing goes for mathematical models. You can make mathematical models of a human to predict how a human will respond to a drug. And so from my end, to me, when we talk about digital tools in biotech, solving the model problem, to me, that's like the big one, is that imagine an in silico human. So this is a in silico meaning on silicon. In other words, it is on the computer. It is a fully digital twin of a person, full person, kidneys, heart, brain, anatomically, physiologically the same as a person. It's a beautiful idea. It's huge. And it's very likely it won't happen in my lifetime. The one case that could actually speed it up is AI. AI may be able to speed it up to the point where we may get that, where it really is representative of a person. So from my end, digital tools, that's what we're going for. And so now thinking about kidney, okay, there are new biomarkers coming out that are in the urine. I'm doing the data work for that to look at how the kidney is doing. So now there's proteins that come out in the urine that you can measure that will give you an understanding of what parts of the kidney are injured. and what parts are not injured. With that data, feasibly, you could create a virtual version of a patient's kidney and then predict, okay, if I give that virtual twin of the kidney a drug, how will it respond? And how will those biomarkers change? So in my mind, that is the excitement, is that you use digital tools of all kinds, of which AI is simply one, but like mathematical models, statistical models, like there's lots of work there. You're trying to speed up that drug development process. You're trying to solve the model problem. And I think that AI can also be used as like icing on the cake to speed up those other digital tools. So you have a digital tool that doesn't use AI, you put AI into it, speeds it up, and then you can solve that problem faster, which then ultimately gets drugs out faster. So when
Yeah,
people
speed
talk
is
about
king.
AI and all that,
Yeah,
to me, it speeds things up. That's the purpose of AI, is that it
I
speeds things
agree.
up, you
Totally
know?
aligned.
Yeah.
Totally aligned. I am definitely on the side of the fence of
Yeah.
it's an accelerator
Yeah.
of what you already know,
Yeah.
and it's not a creator.
Not
It's not,
really.
that's where the AI slop is.
Yeah,
But
and I mean that said, right? So like thinking of like generative AI, like LLMs and all this stuff, ChatGPT, all the new AI models, those are being employed in drug discovery because what they're doing is they're saying, what are all the possible drugs that we could possibly throw at this receptor to see what is going to happen? Well, an AI system is great at that because an AI system can just say, well, yeah, here's all these different ideas, right? A generative AI system can come up with those things. But then as soon as you have that compound, now you try to develop it. You try to develop it into an actual drug. And AI doesn't know what drugs are going to be manufactured the easiest. Like it has no idea. It would need data specific to that exact problem to then be able to look at it. So yeah, so I did just want to kind of put a caveat, like it is creative, and it is used that way creatively, but in general, it's an accelerator. And that's the way I look at it because I'm not building drugs. That's not what I do. What I do is I see how is a drug going to affect the kidney once I know what the drug does.
gosh, okay, so with digital tools, AI, and where things are going, it sounds like it's pretty possible, or it possible in the sense of like how much can you actually hit certain milestones?
Yeah.
Because
Right.
technically, like in the research and development world, I mean, you're really embarking on a mission here that, I mean, you want the number to go down, right?
Yeah.
And it sounds like what I'm
Right.
hearing is like everything seems like in parallel. So, like, even if the research about knowing all of these, like, you know how you mentioned about aneurysms, right?
Yeah. Right.
It's like, okay, all these kind of mysteries that are happening
Right.
because it isn't exactly well, there's not enough emphasis on it in a way. But then also with the drugs, it's like, well, if people know that they have kidney disease, then what are we going to do with them?
Right.
Because they're not going to have a solution because there is no real solution
Right.
pre the dialysis side of things,
Right.
and then
Right.
until the kidney actually fails. So that's not exactly helpful. And it's kind of like putting a seatbelt on before knowing you're going to get into a car accident, right? And it's like,
Yeah.
okay, what am I going to do when I'm actually in the car accident? It's like
Right.
you don't want to
So
wish for that.
yeah, so I'll give you a really good example. So
Yeah.
acute kidney injury
Okay.
happens in 1%, and so I may be getting this wrong, but hear me out because I know it's along these lines. 1% of all hospital patients get acute kidney injury. 1% of all hospital patients. Now, that's a lot of people. So, acute kidney injury, the thing is, we've made, different systems that predict what patients will get acute kidney injury. We can predict actually, and a really good example, Jay Coyner is at University of Chicago, Perry Wilson at Yale. wonderful guy. Both of them, wonderful guys. They have developed very sophisticated machine learning algorithms that predict what patients will get acute kidney injury. Now, why is it that these aren't used in hospitals? They may be in small cohorts, they may be using those algorithms, but a big reason that they don't use them clinically is because just because you know who's going to get acute kidney injury doesn't mean you can do anything about it. There's no drugs they can give them. There's no, you know, maybe there are certain things like, oh, we're giving them this drug. Well, maybe we shouldn't give them this drug. Maybe we should give them this drug because, but in the end, like they don't actually have actionable problems that this AI system, this machine learning system, this mathematical model, whatever this digital tool is, predicts will happen. They can't do anything with that information. And
Right.
so this is this challenge with kidney care is it's like I can predict that you're going to have a problem, but there's not really anything I can do. Now, if it was chronic kidney disease, so it's like, oh, okay, we look at your genes, we look at your weight, your age, are you male, female, assigned at birth, whatever. The and maybe they could say, okay, you are at risk of developing chronic kidney disease, so you should follow this diet. And maybe this diet, if you are at higher risk of chronic kidney disease, the diet becomes more intense. Like there's this idea like that could maybe work, but chronic kidney disease develops over the course of decades. So
Mm.
like that's not a product that gets sold. You can't say, "Oh, you're at risk, here's this app that you paid me that you're paying me $5 a month." It's like, "Well, yeah, just because it says you might have disease in the future and that you should follow a diet." It's like, "Well, why not just say that that everybody should just follow this diet?" Like
Right.
what's,
It's like Weight
you know?
Watchers.
So
Like,
yeah, so it's complicated. It is very
yeah.
there's a lot of chicken or egg problem that
Yeah.
that you run into, and so this is what I think about, day in, day out. This is what I wake up thinking about is how can we actually address this? How can we actually address real substantial problems that can be solved? How can we prevent someone from developing acute kidney injury? You can predict it, but can you prevent it?
Mm.
So, and it's tough because if you get it right, you can change lives and you can make some money. And that's great. But it's hard to get it right on this one. It's really tough. And so a lot of my job is just talking to people to figure out, well, what do you think the problem is? What do you think the problem is? What do you think is the solution? And in the end, I think things will shake out and there will be opportunities that will become available. But it's hard. It's a hard space, and it takes a lot of time and a lot of conversation to figure out kind of where these angles are.
Right. And wow, I learned so much because this isn't just like humans, as in like with just failures. This is
Yeah,
also people like you're saying that are maybe know more about diabetes, right? Because
yep,
that's a little bit more mainstream, or
totally.
high blood pressure, like you're saying, or
Or they
heart
have
disease.
cancer. They like have
That
cancer
too.
and kidney disease and diabetes
Oh
and
my
heart
God.
disease. That
Oh,
explain I mean that explains many people. Like many people are in that scenario. They have some kind of
yeah.
cancer, maybe they need chemo. The chemo hurts the kidney, and then that cycle continues. So
And
it
not
is
only that,
very
humans, but also
complex.
pets too, right? So it's like,
Yeah.
wow.
So it's funny because, you can talk about this in one way and it makes you feel like all the solutions are possible. And then you can talk about it in another way and it feels like the problem is completely insurmountable. So it's somehow we find a balance in that. I find myself going to both places all the time, seemingly oscillating between the two. But somehow we keep going forward. I think that's a pretty typical experience for scientists, especially biomedical, is it's like You know there's a solution, but you absolutely can't see any solution at all at the same time. Crazy.
If we were to wrap this up in a way to summarize, what would you say is your zone of genius, Owen?
Oh, my zone of genius. Constraints. Working within a box. So I need to know where the limits on a problem are. I need to know what the boundary looks like. And then I'm there. And my ability to work with constraints, I think, is the creative piece that I bring. So if I'm a genius, which I don't think I am, but if I was, I would say that's probably my area of genius, is constraints.
Such a smart response. I'm going to be
thank
thinking
you.
about that for some days now. And be like, okay. It's so interesting because structure does help you to
Huge.
want to think outside the box.
Yeah, totally. You got to know what the box is before you think outside of it. Yeah, big time.
Owen, thank you so much for being very generous with your time.
Oh, my pleasure.
And wow, I learned so much about you. I learned so much about the incredible camaraderie that you have. Basically went on an incredible journey in getting to where you are today. And I am so excited because technically you are top 5 kidney researchers in the world.
I'm top
Is that
5
right?
kidney math researchers in the world. There's
Kidney
better
math
kidney researchers
researcher.
than me, but kidney math, I'm top 5.
Amazing. Okay, well, I hope you keep on going because I kind of really dig the whole how to be objective and de-risking
Yeah.
through
right.
math.
Totally.
And in the space and environment as a scientist, to be able to experiment and then being able to make something out of it as an engineer. So
Yeah.
I'm very excited. Oh my gosh, that was awesome. Thank you, Owen.
Thank you, Cee. This
you.
has been great. Thanks.
Amazing. Thank you.